Hello, this is Peter Kritsch, and I'm going to tell you a little bit about Biotechnology 1, which I believe you're seeing during 6th hour. Grade level for Biotechnology 1, primarily juniors and seniors, though there are a few sophomores who are in Biotechnology 1 as well. The overall goal, purpose, and priorities of this particular class, the goal of Biotechnology 1 is to introduce students to how DNA is used as a tool in genetic engineering, genetic identification, and genetic testing. So students should be able to get a lot of hands-on experience to be able to analyze those results, move them, watch how DNA is moved from one location to another, figure out how we learn about its sequence, and a variety of things. All the different hands-on activities that we get to do, all the labs that we get to do are recorded in their lab books, and I do let them use that on the lab book part of the test. And so they're practicing not only analyzing their experimental results, but communicating their findings to me to some extent, but mostly important to them. So that whatever they're writing down, whatever analysis they're doing, is something that they can understand as they move forward in the class. And lastly, not only do we talk about the science behind the concept, the technology itself, and how that technology is applied, but we also take a look at whether that technology should be used in that way or not. And so we take a look at bioethical issues such as cloning, stem cell research, genetically modified foods, in addition to whether they should be labeled or not, genetic testing, and a wide variety of other situations where students who, even if they don't decide to go on into a career in science, they'll still be asked to make some decisions about each of these. So a chance to kind of practice that. Where we've been so far in the class. We started off by taking a look at what biotech is and getting some background idea about that concept, and then we focused in on cellular respiration as a tool for making foods such as root beer, yogurt, and cheese, but also fuel such as ethanol. We also looked at DNA structure and extracted DNA from cells. Second unit, then we really start to dive into the genetic engineering side of biotech. And they do a heat shock transformation experiment where they take DNA from a few different sources, including jellyfish that biofluoresce, so they glow under UV light. And we take that protein from that jellyfish, transfer the gene for that protein into bacteria, and get the bacteria to do the same. Students also learn about gene therapy as an application of that, and then we introduce them to positive and negative controls. In the third unit, we look at genetically modified foods. They bring in a food sample, we extract the DNA, analyze it using a process called polymerase chain reaction, and analyze the results of that PCR by gel electrophoresis. In our fourth unit, they're learning about genetic testing using PCR again, but they're looking at a sample of their own DNA this time. Where we're going next, after this, we do a short 2-week unit on cloning and stem cells, learning about how cloning has been used for sheep and a variety of other organisms. Why would people want to clone? We also take a look at embryonic stem cells, adult stem cells, and a new source of stem cells that may eliminate the entire ethical debate in the first place. And then lastly, we spend the last 3 weeks or so of class doing a semester lab exam project, where students will take everything that they've learned this semester and really kind of put it together. They'll extract DNA from themselves and end up creating a bacterial-humid hybrid. Specifics of Thursday's lesson. Where students are in the middle of this PCR process. They've extracted DNA from their own cheek cells, but they're going to be setting up a PCR to help determine which versions of a bitter taste receptor gene they have inherited. Long story short, everybody has 2 copies of each gene. This particular gene allows you to taste a bitter chemical called PTC. That bitter chemical can be found in broccoli and Brussels sprouts and a wide variety of other type of leafy green vegetables. And people have either inherited 2 versions that work, so they are strong tasters, 2 versions that don't work, and they are non-tasters, or one of each, in which case they are weak tasters. We're doing this particular approach because it doesn't involve a genetic disorder. We're not looking for anything like risk for cancer But at the same time, the process is exactly the same as what would be done in the lab. So we can detect whether someone is a carrier of that non-tasting gene, even though they themselves might be able to taste that bitter chemical. In addition to setting up the PCR reaction, they're going to be listening to and responding to a genetic testing situation. To back up for a moment, again, remember, students have set up a PCR before, so the process that you're going to see today really shouldn't take them too long. The first time through it definitely did, but the second time through we're able to move a lot quicker. They know what they're setting up, and so we should be able to get to that genetic testing situation in the last third of class. Looking forward to your visit, and looking forward to your questions and insights.